Welcome back, guys, to part 2 out of 9 for the second season, I don't even know if I can call it that, of the biology lecture series. Today's topic is about the aerobic production of ATP. I love this lecture. Okay, we learned in the series of lectures for the first exam about glycolysis, which is essentially the first step of aerobic ATP production. So make sure you understand glycolysis relatively well, in that you know that 2 net ATP are produced with 2 NADH and 2 pyruvates per glucose molecule. Now, glycolysis itself is an anaerobic process, meaning no oxygen is involved, but through aerobic respiration, we can turn 1 molecule of glycolysis into 36 ATPs. Here's how. Everyone can recite that the mitochondria is the powerhouse of the cell in their sleep at this point, but why? The mitochondria is known as the powerhouse because it is home to the TCA cycle / citric acid cycle / Krebs cycle and the electron transport chain. Don't ask me why there's so many names for the Krebs cycle. I don't know, science is crazy. Now, before we talk about the specific processes of aerobic respiration, I would like to touch on the symbiotic origin of the mitochondria. So it's actually stated that long ago, the mitochondria was a bacterium that was engulfed in an ancestral eukaryotic cell that just ended up having such a fantastic relationship with the eukaryotic cell that it became arguably our most important organelle. Now, that's a personal opinion. Do not quote me on that. I just love the mitochondria. It's such a good organelle. Okay, enough of that. Let's talk about how we get so much ATP from 1 glucose molecule thanks to the mitochondria. All right, after glycolysis in the cytosol, we have our product pyruvate. You do need to know the structure of pyruvate. Pyruvate gets shuttled to the mitochondrial matrix, where it gets converted to acetyl-CoA through an enzyme called pyruvate dehydrogenase. Okay, you also have to know the molecules in this reaction and how to got to be able to recognize the structures. This reaction generates 1 NADH, which is an electron carrier molecule that is used later on in the electron transport chain. Acetyl-CoA is a strong energy carrier, and this reacts with citrate synthase and oxaloacetate to form the very first product of the TCA cycle, the 6-carbon molecule citrate. And as you may have guessed, you also have to know how to draw this too. I'm sorry. Luckily, that's the end of all the drawing memorization you have to do. We don't require you to know each individual step of the TCA cycle, but if you're curious, it is oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and then back to oxaloacetate. So 1 round of the TCA cycle overall produces 3 NADH and 1 FADH2. Remember, those are our electron carriers. But remember, 1 glucose molecule produces 2 pyruvate. So per glucose molecule, we actually have to run through this entire process twice. So in total, the TCA cycle produces 6 NADH and 2 FADH2. Common misconception, we do not get the majority of our ATP directly from the TCA cycle. We get our ATP from the electron transport chain located in the mitochondria's cristae. This process is called oxidative phosphorylation. Here we have my very crude drawing of the electron transport chain. Let's walk through the steps together. So step 1, NADH is oxidized at complex 1. This drives 4 hydrogens across the membrane, and the electrons go to this, and then into complex 3. Complex 3 is just another further reduction in energy. While NADH is being oxidized at complex 1, FADH is oxidized at complex 2. And then those electrons are also moved to complex 3 before they go across a cytochrome C bridge, which is another electron carrier, before hitting complex 4. At complex 4, electrons interact with the final electron acceptor, oxygen, to produce water. Please remember that the final electron acceptor is oxygen. So Approximately 10 protons are pumped across the inner membrane against the concentration gradient as a pair of electrons are transported from NADH to oxygen. Let me repeat that. Approximately 10 protons are pumped across the inner membrane against the concentration gradient as a pair of electrons are transported from NADH to O2. All right, 10 protons per pair of electrons. All right, good. Finally, there is ATP synthase at the end of all of this, which is technically electron transport chain complex 5, but I'm just going to call it ATP synthase. This is where those hydrogens in the intermembrane space enter the ATP synthase complex and undergo a certain merry-go-round motion thing. Each hydrogen occupies a subset of the ATP unit, and that gives the ATP synthase rotation energy, which drives the energy of ATP formation. So it's that rotation energy that gets us from ADP to ATP. I think that's so freaking cool. So that's all for this video about aerobic respiration. As always, I will have my disclaimer at the end of the video. Number 1, I'm a student, not a professor. Number 2, my biology knowledge is by no means perfect. And number 3, I'm making this series using my college notes. If you find an error, please let me know. I love criticism and I don't want to be sharing wrong information with you guys. I hope this video helped in some way, and see you next time for our next video on photosynthesis. Bye!